We'll start our program right away. Dr. Gupta is already with us. He is just checking out his presentation. We'll start with, we requested all to wait for some time, so please hold on. Yeah, it's loading. Yes, yes. You may try full screen. Yes, it's fine. It's fine. It's coming. It's coming. That's nice. Everything is all right then. Just 2 more minutes, we'll start. Okay. Joel, are you there online?
Yes, sir, I'm here.
Everything is all right. So is it good to start?
Yeah, obviously, we can start now.
Streams checked?
Yeah, everything is checked and it's working perfectly fine.
Okay.
Yeah.
So in a minute or two, we can begin and you can start recording and going live, right?
Yeah, sure, sir.
Okay. Dr. Gupta, is it all right at your end? Can we start? You might be muted. We can't hear you.
Yes, everything's fine at this end.
Okay. So we are going.
Okay. Well,
Okay.
thank you for the invitation to speak. And I'm going to give a.
I'll begin. I'll introduce you and then
Oh,
I'll
sure.
call you
Okay.
up.
Sure.
Okay. Just give me a few minutes. Welcome, viewers. Welcome to the session or to this session of live webinar from Regional Science Center and Planetarium Calicut. You all know that we are part of the National Council of Science Museums, which is the single largest network of science centers. And we have about 25 science centers at our fold. And apart from that, we also support another 30 odd science museums all over the country. We also have a few science museums and other sort of museums built by us but supported by other organizations outside India. Anyway, but this is today's session, and in today's session we are going to talk about the variants of concern regarding the SARS-CoV-2. You all know, since the beginning of this pandemic, we have tried to reach out to the experts who are directly connected to the research and subsequent remedy of this pandemic. And today we have one such session, and I'm so very excited to introduce today's speaker over here. Let me just narrate the situation a little bit. Since the SARS-CoV-2 variant known as B1617 appeared and first reported in India, there was an increasing interest about the variants, the SARS-CoV-2 variants, and we came to know all about it. We got concerned about it, and of course, the scientists, they were also uh, delving into the details of this transformation, this mutation, and all. And already we know that in several countries these variants have spread, and in some regions they have also become the dominant variant for the infection. Now, since then, probably researchers have found out more variants. We were talking about B1617, but later on there were B16171 and B16172 and B16173. And the key questions, however, remain about how quickly these fast-moving variants can spread, their transmissibility, their infectability. And we have a speaker today who is at the very forefront of the international research into such topics, such subjects, and we have with us Professor Ravindra Kumar Gupta, who was there in the Time magazine list of first 100 most influential persons of 2020. He is a world-renowned virologist whose basic field was initially researching HIV, but now he has also taken into his focus the SARS-CoV-2 since the beginning of the pandemic, and he has produced some major results. He has been concerned about India because on several occasions he has talked about the movement of variants in India. He has talked about the possibility of a third wave, and today we are going to listen to him. Professor Gupta is a professor of clinical microbiology and welcome senior fellow in clinical science at Cambridge Institute of Therapeutic Immunology and Infectious Diseases, Jeffrey Chia Biomedical Center, University of Cambridge. He is honorary consultant physician infectious diseases at the Cambridge University Hospitals NHS Foundation Trust, and he is adjunct faculty Africa Health Research Institute, South Africa. He is honorary professor University of KwaZulu. I don't know whether I pronounced it correctly, and it is in South Africa once again. And he is a person who has always been a major researcher in virology. And today we are so very happy to get you here, sir. We are honored to host Professor Gupta, and it is over to you for your talk.
Thank you very much for that kind introduction, and very pleased to be giving this talk to colleagues in India. So the talk is going to be about the SARS-CoV-2 variants, and in particular Delta variant, and what the implications are for spread and control with vaccines. So here's SARS-CoV-2 diversity since the beginning of the pandemic. You can see down in the left-hand corner the 19A lineage, down here the Wuhan 1 strain, and how this has expanded over time. And in the colors are the major variants of concern, or VOX, with the old and some of the new nomenclature. You can see here that up here you have the alpha variant in blue, beta here in dark blue, gamma. This is so let me tell you what these are. These are the original UK variant up here that spread from Southeast England, and here we have the virus isolated in South Africa, the beta, and then gamma. This is the Brazil P1 variant, and of course down here you've got Delta, which is the B1.617.2, and then Kappa, which is the 0.1. So a lot of things have happened in the last 18 months. How is that happening is the question, because the virus is not evolving at such a high rate. If you look at just in vitro mutation rate, it's a coronavirus that has a relatively modest mutation rate. The reason this disease is different is because last year we started realizing that chronic infection was a major part of the biology of this virus. We were finding people shedding virus for long periods of time. It's probably less than 1 in 20 people, but it's still significant. And in particular, people with immune-compromised bodies who are on immune suppression. For example, this case we described in Nature earlier this year, where somebody with lymphoma, currently treated with rituximab, a B-cell depleting agent, which obviously lowers your antibodies and makes you susceptible to infections. This individual was infected, as you can see here on the right side, over a number of days, 100 days here, in fact, until he unfortunately died. And this is 3 months of ongoing infection. The reason was, of course, his antibody levels were low. And here we've shown some mutations that are arising just in the spike region. And we're showing the prevalence of the mutation in the sample. So we're using deep sequencing. So we're looking at how much of the virus contained in that sample has different mutations. And you can see here that the virus for the first 66 days is pretty much identical to the one that was infecting the patient. So very little change is happening in the spike region until we start treating the person with convalescent plasma, which contains neutralizing antibodies. And convalescent plasma was used a lot in India, I think. The problem with convalescent plasma is that you can't measure very accurately because each unit of plasma is from a different patient who has recovered from infection. Each person may have different levels of antibodies, which depend on many factors. So the potency of the antibodies is variable. And you can see here what happened in the patient, which is that after administering this plasma, when we next sampled the patient at day 82, around 16 days later, we can identify this new population of viruses in the patient, characterized by a deletion in the spike protein as well as another mutation. And then, interestingly, that new virus disappeared for some weeks until we gave another unit of plasma here at day 95. And these new population of viruses, we don't know why these came up, because the antibodies will have disappeared by the time the antibodies last around 2 weeks in the patient's blood. So we obviously disturbed the equilibrium. We generated some new viruses, new variants. But then after the third plasma infusion, these original ones came back, suggesting to us that this plasma is selecting through Darwinian selection virus populations which are relatively resistant to those antibodies in the plasma. And that was a hypothesis. We then tested that in vitro, and we found indeed that this D796H mutation made the virus a little bit resistant to the antibodies in the convalescent plasma. This mutation, Delta 6970, which was the other mutation coming along at the same time, is now known to be in a number of different variants, including the alpha. And we showed in vitro in 2 papers that this increases the infectivity of the virus. It makes it more infectious, and this is another, and this is potentially a compensatory mechanism for this mutation, because this mutation allows escape from antibodies, but it comes with a fitness cost to the virus. The virus is not so able to replicate when it takes on this mutation. And so what this one was doing is restoring that replication defect. If you look here, this is looking at more mutations across the whole genome of the virus, the 30 kilobases which are there, and not just the spike protein. And you can see the key thing is that there are obviously different mutations happening here, including the ones I just talked about in S, the spike protein, here. But you can see these NSP protein mutations in other genes of the virus. But these groups of mutations come together and disappear together. And you can see another group here. And what this is telling you is that there are virus populations, groups of viruses which are coming and going and competing with each other in the patient. And this is one of the first descriptions, really, of the kind of complexity of genetics that can happen within individuals who are infected with SARS-CoV-2. It's not just a single variant that continues without any mutation. What we're showing is when you start playing around with by infusing antibodies, and in a person with very low immunity, you can generate a lot of virus diversification. And this really is the blueprint, what we are showing here for the variants, because you can see the variants have many mutations. And this is a very good explanation of how that happens. So I've shown you that mutations can make people resistant to the antibodies in convalescent plasma. And this is an important concept because, of course, vaccines are, when you take a vaccine, your body makes antibodies against the virus, and the levels of antibodies and the type of antibody and the type of vaccine is critical. This figure on the left shows you different types of vaccines and the observed efficacy versus the in vitro efficacy of those vaccines. And you can see that the clinical efficacy reflects very nicely what you predict in vitro from neutralization assays. And the mRNA vaccines seem to do the best in this sort of analysis. Another piece of data or evidence showing that neutralization of viruses in vitro is important. And essentially, to do a neutralization test, you take virus, you make a virus with mutations, maybe or a wild-type virus. You then mix it with antibodies or serum from people who've been vaccinated, and then you test the ability of those viruses to cause infection in new target cells. And if you block those infections, that's neutralization, and that's what you're looking for because the antibodies in the blood are blocking that virus from infection. And this is an analysis from the some of the Chaddocks, the group in Oxford, where they have done in vitro, which is through in vitro testing of neutralization using pseudovirus testing. And they've looked at the risk in patients. So they take the blood from people who have been vaccinated, then they look forward to see who gets infected. And they can correlate that back with the amount of antibody these people had. And they find that if you have a neutralization level of around sort of 185, then you're well protected. But if your neutralization response or your antibody response is poor, then you're much more likely to get an infection up here. The relative risk goes up of infection. And so, and of course, the higher your neutralization antibody response is, the lower your risk down here. You can see the line is going down. So for reasonable protection, you can see here a value. And this is important because it tells you when you make a vaccine, you can test how good the vaccine is, not by even waiting for people to be infected, but you can just look at the amount of virus antibody produced and make predictions on that. This is complicated by the fact that different people make different levels of antibodies. You can see here that age has a big impact on how well you neutralize virus. You can see this is a paper we published in Nature this year. Again, another paper. So this is the probability of a person having detectable neutralization as you become older. And of course, for younger individuals, you have very high chance of making antibodies after Pfizer mRNA vaccination. But after the age of 80, the probability drops off very sharply. So this is obviously a risk group of people. And if you look at the variants, then people are much more susceptible to the variants. They're more vulnerable to variants if they're over 80 years old. This is the probability of having no protection from antibodies. It becomes quite high with the variants compared to the non-variants in the elderly age group. So let's move on to the Delta variant, because we've talked a little bit about how variants emerge. I am not going to spend time talking about alpha, beta, gamma variants because I think people know a bit about them. But the Delta variant, of course, is topical because it emerged in India, we think. And this is some data from a paper we published earlier this year in collaboration with colleagues in India from INSACOG. And it shows you essentially the group of viruses which are the 617 lineage, as my colleague earlier described. And this lineage is grouped into 0.1, 0.2, and 0.3, which is up here. I haven't labeled it. These viruses have come from an ancestor. We don't know where this emerged. It was probably somewhere in Western India, but it could have been elsewhere. And it differentiates itself from other viruses with the L452R mutation, which are in other parts of the world. For example, the California variant, which had emerged, and of course that virus had the Y144 mutation. You can see here these are different mutations along with the different lineages. The Delta variant here is characterized by the T748K mutation in the RBD, receptor binding domain, and it has L452R, of course, and it has this deletion in the N-terminal domain. The 0.1 variant I'll talk about a bit later, which was originally called a double variant, the double mutant in India, which created quite a lot of concern, has an E484Q mutation, which is associated at a position that we were very worried about in terms of escape from antibodies. This is a graph that shows you how the proportion of variants or viruses has changed over time. You can see here the rise in green of the 617 group of viruses here since February this year, and now dominant is this light green one, which is this one here, this 0.2 or the Delta variant. There is a mix, of course. The 0.1 variant was initially dominant and has now been outcompeted, we believe, to a large extent by the Delta variant. And of course, we need to know why that is. Here's a bit of basic biology about the spike protein. You've got, of course, the virus here with the spike proteins on the surface decorating the virus. It's a complex protein made up of 2 subunits, which then interacts with ACE2. The ACE2 it's actually an enzyme which is present on our cell surface. And this is the blow-up picture showing you that the spike protein actually is processed into these 2 components, the S1 and S2. The S1 contains the receptor binding domain, which interacts directly with ACE2. And if you blow that piece up, you can see here some of the molecular sort of details of the cell up here with ACE2 and the virus down here with the spike protein. Here are some of the key residues that interact between the ACE2 protein and the virus spike protein. E484 I mentioned earlier. A change to K in this position is present in the Brazilian and the South African variants, or the beta and gamma, and is mutated in the to the Q in the 617.1 virus that I described earlier. So the 0.1 virus that was dubbed by some of the Indian press as the double mutant. This was sort of earlier this year, something like February, I think. And when we heard this, we were interested, and we saw, we wanted to know whether this double mutant really was something to worry about, because the L452R mutation was well known from the California variant as conferring a degree of immune escape and increased ACE2 binding. But we didn't know what this Q at position 484 was going to do, because it had never really been tested. So this is showing you where those mutations are on the spike protein here. They're up here in the receptor binding domain, both of them near to each other. And this is how close they are to a neutralizing antibody that's been solved, the structure of which has been solved. This is the H4 neutralizing antibody that interacts with the RBD. And you can see here that the E484 is right there at the interface. And so changing this mutation, changing this amino acid, will impact the binding of this neutralizing antibody. Of course, we make many different neutralizing antibodies. This is just one example. So what we're showing you here is a group of different mutations made in artificial viruses and the amount of neutralization. In other words, how these mutations affect the ability of serum containing vaccine generated antibodies to neutralize. And so the wild type, the Wuhan 1 virus, is here, and you can see that all of these mutants have a roughly 4 to 5 times reduced sensitivity to those antibodies. In other words, they're a little bit resistant to the virus, to the antibodies. But it doesn't matter whether you have the double mutant here, which is in the Indian variant, or in fact in a triple mutant with the additional mutation seen in the 0.1, as compared to the E484K alone or the Q alone. So what we're saying here is that the double mutant here does not really seem to have a dramatic impact on the neutralization. So moving on to the Delta, this is the 0.2 virus. What we're doing here is showing you a blow-up picture of the spike protein. And this little diagram shows you some of the key mutations in the Delta variant compared to the Wuhan 1 virus. You can see there are quite a few differences in various parts, including the receptor binding domain, which I described to you before, the L452R and the T478K. There's also key mutations in the cleavage site, which is where the spike protein is chopped in two, and it needs to be cleaved or chopped in order to be fully infectious. And so any mutations in this area have been shown to facilitate that cleavage, that infection process because of cleavage. And this is the cleavage region here in white, the white box, and it's blown up down here, showing you the 681R mutation. We think this is very, very significant for the virus. So here's the 0.2. I showed you there's some structures of the 0.1. This is the 0.2 virus structure has been solved in relation to one of the Regeneron monoclonal antibodies that was produced in order to try to design new therapeutics. And you can see that indeed the 478 potentially is an interacting site with the Regeneron antibody, and therefore may affect binding of that monoclonal in particular. This is a graph just showing you how the number of cases of different variants has changed over time. And you can see here the rise of the black bar here, the B617.2 Delta variant, in conjunction with the 117, the UK Alpha variant. If you were to look at more recent data, then there's very little Delta B117, and it's mainly Delta now. Globally, this has also been recapitulated with the Delta taking over. In terms of infectivity, this is the other big question. Why is Delta being so successful? Well, I've shown you some data on the epidemiology of its expansion. If we test these live viruses in the lab, we infect lung cells here. These are lung cells that are growing, and you can infect them with SARS-CoV-2 Delta. We compared the alpha variant 117 versus the beta variant here, the Delta variant, and you can see the black bar here, for example, at 8 hours post-infection is very much higher. This is a log scale, so it's very, very much higher, like 1,000 or 10,000-fold higher for Delta than it is for alpha. And if you look at 24 hours, there's around 100-fold difference. So still very significant differences between the two viruses. And you can see here measured in a different way. You're also seeing very, very significant differences between the alpha variant down here and the Delta variant. So this tells us why in India, the B117 alpha variant has been taken over by the Delta variant. If you look in human airway epithelial systems, which is it's a more complex model system, you see a similar difference between B117 and 617.2, or the Delta. The green bars are generally higher than the orange bars here all the way across, telling us that there's higher infectivity. So the virus is more infectious, it transmits faster, we can see that in vitro as well as in people. What about evasion of the immune system? I showed you some mutation data earlier, but now I'm going to show you some live virus neutralization data. So we're mixing Pfizer vaccine sera here and ChAdOx1 or Covishield sera here. We mix those sera with live virus, and we check the ability of those antibodies in the serum to block the virus from causing infection in the lab. And you can see that Delta is more resistant to the serum for ChAdOx1, and it's more resistant to the serum from Pfizer vaccinees as compared to wild type or the B117 virus. So you can see the green dots are much lower down here in both cases. If we use a different type of virus system, you get the same result. So Delta is less sensitive to neutralizing antibodies. It explains why in India we've seen people becoming reinfected, because they were infected originally with the wild type virus here, and you can clearly see there's a difference now. The virus is less sensitive to those antibodies, and so that's why you're getting reinfection most likely. Well, what about the monoclonal antibodies that have been made for treatment of SARS-CoV-2? Now, these are not available in most countries yet. They are available. They have been made in the US mainly. They have been undergone clinical trials, and certainly the Regeneron cocktail in the recovery trial in the UK was associated with a mortality improvement. So the survival advantage, there was a survival advantage if treated early with the Regeneron cocktail of these two antibodies here. But these clinical trials were done in patients infected with the original strains of the virus, not with the Delta. So we have done some work to look at whether the Delta variant is less sensitive. And indeed, if you look at this, because one of the monoclonals, casirivimab, there's no difference between the black and the blue. So that shows you that the Delta compared to wild type is pretty much the same. But for this imdevimab monoclonal antibody, there does seem to be a separation of the curves. In other words, the Delta may be less sensitive. So we are a bit worried about using the Regeneron cocktail for treating people with Delta infection. Similarly with the Lilly cocktail, this is a different set of antibodies. You can see that there is a significant loss of activity against the Delta variant. And this antibody is already known to be less effective against the beta variant in South Africa. So combination monoclonal antibodies may need to be improved in order to deal with the new variants, and that's another take-home message. Another important observation we've made is that, why did the Kappa variant, the B617.1, how was it outcompeted by the 0.2 or Delta? Why did one emerge first and then largely kind of fade away? And the reason we believe is because the Delta is more infectious. Here you can see the comparison between 0.1 and 0.2. The bars here are higher than the wild type down here, as well as the 0.1 virus. So the Delta seems to have an advantage even over its cousin, the B617.1 or Kappa. And same in these replication assays in vitro, we compare the two viruses. You can see at different time points after infection, this bar is much higher. So the 0.1 is outcompeted by 0.2, which explains what's going on. This is just a slide showing you what's happening in different cell lines and different types of target cells. It's pretty much the same story. And then the other intriguing area of the biology of Delta is this thing called syncytia formation. Now, it's been known for some time that SARS can join cells together, fuse cells together. And the wild type is shown down here, the Wuhan. Where the cells are, where the fusion has occurred, the cells are going green. And this assay or system of analysis is using something called green fluorescent protein, GFP. And when two cells come together to fuse like this, you get the green. If there's one cell expressing spike, it doesn't go green until the fusion between these two cells has occurred. Delta, you can see, generates a lot more green than the wild type. And that is very important. This is a quantification of that ability of Delta to generate syncytia of cell fusion. You can see that at early time points, it's much more efficient in generating those syncytia. So you get a lot more green confluent cell area. That potentially links to pathogenesis and viral load. We don't understand that fully yet. So what's the implication of all of this? I've shown you evasion of antibodies from vaccine. I've shown you some data on infectivity of the virus. And really the translatable importance of this is shown very nicely in analysis we have done in collaboration with colleagues in India in 3 hospitals in Delhi, where they documented very nicely healthcare worker-associated infection in people who are vaccinated. You can see this is a phylogenetic tree, just showing you the relationships between each dot is a person who is infected, and the green ones are the Delta infected patient in healthcare workers, and these other colors are different strains, which are, for example, B117 and the 0.1 Kappa variant here. You can see that there was this group of very closely related green dots, both CT values, which is the amount of virus in the sample being quite large. And they have very similar sequences. This is indicative of a super spreader event where one person infected a number of individuals, all of whom who had 2 vaccinations. This is dark blue, shows that they had 2 vaccines. 1 or 2 of them had only 1 vaccine, but largely these were double vaccinated people with a good time period between the vaccine and being infected. This is the number of days since vaccination down up here. And you can see here, this is the dates at which they were infected or diagnosed. So this is quite worrying because, of course, you're getting infections in people who have been vaccinated. They have significant viral loads, and we predict that they may be able to pass this virus on to other people. And that would be a worrying situation, a breakthrough of a vaccine as well as transmission. All of these were vaccinated with Covishield. Of course, none of them had severe disease and none died, which is the major take-home message. You get mild to moderate illness, but you can transmit. Now, if this virus is transmitting, it's possible that it's still evolving. And this is a figure here, a bit complicated, but it's essentially showing you in green the number of mutations which are emerging on top of Delta in small numbers of people, right? But still, this shows you that Delta is not stationary. It's still moving. In other words, we still need to chase it potentially. And these mutations, some of them will confer additional loss of sensitivity to neutralizing antibodies. So our antibodies from vaccine may be becoming less effective over time. So we really need to drive down the transmissions to drive down the evolutionary advantage of that virus. So what about a possibility of the third wave in India? Well, there were a large number of infections in the first quarter of 2021. There will be a proportion of immunity to Delta, but of course, immunity does not last forever. And we can see from data that after 6 months, some immunity starts disappearing. In other words, you can start becoming reinfected with the same strain potentially. And we've seen with the lifting of restrictions such as lockdowns that the number of cases in India is rising, in particular in Kerala and other parts. This is likely to be a slow wave at national level that is not as severe, but it will still put pressure on health services, I believe. And we need to watch for additional variants or mutations, as I showed you in the last graph, modified versions of Delta that may make this an even more difficult thing to control. So what about the future of vaccines? We need Delta-specific vaccine, I believe. I hope that manufacturers are taking note. We may need what we call a multivalent vaccine, which is a mixture of different strains of the virus. We need better equity in terms of access to our vaccines, and we need to be able to test our vaccines efficiently so that different vaccine platforms or manufacturers can be compared to get the best vaccine for a particular population. Finally, boosting is probably going to be needed. In other words, the elderly, people with bad responses or poor responses to the vaccine in the first round. For example, diabetics may not respond so well, and people with other risk factors. And then finally, we really are looking to the future here to pan-coronavirus vaccines. And what this figure is showing you is that some groups in the US have now been able to identify, this is a spike structure, for example, showing you in blue the areas which show a lack of evolution across the globe, and areas that show lots of evolution. So the receptor binding domain is up here. You can see there's more variability towards the red end of the spectrum. So we're not going to design immunogens against red areas. We're designing designing vaccines against conserved areas which don't change. If you can do that, then there's less chance for vaccines or the virus to escape vaccines. And so there's some very nice work going on for the future of coronavirus vaccines. Here's a slide just showing you the wonderful people we've worked with in order to bring this information to you. And I would like to answer a few questions if there are any. Thank you.
Thank you very much. Now I'll invite questions from the viewers. You can type them in the chat box, or if you so wish, you can unmute yourself and ask your question. But by the time the viewers come out with their questions, Dr. Gupta, I have an interesting case today that has occurred in my institution. It has been recorded today itself. A person who had both doses, who had one infection, he has been reinfected here, and today he has been declared as positive once again. Do you think that this can be the cause of the Delta or a particular variant that is working otherwise?
Yes, I mean, so the data we showed you that if you can be infected even because of after a vaccine, that reinfection is also possible. We did an analysis of the epidemiology of the growth of Delta in India, and it was consistent with people being reinfected. And of course, there are data coming out that in India there has been a significant degree of reinfection of individuals. So yes, that is entirely possible.
Is there any study that if at all there is any reinfection happening, then would there be any change in severity? Can we expect something otherwise?
Infectivity, yes. Delta's infectivity is very high. So even with some immunity, it can infect you and it can be passed on. As we showed you in that slide from the healthcare workers, that is a distinct possibility.
There we have a question from one of our viewers, and he's asking from North Bengal Science Center, Siliguri. He's asking that could new COVID-19 variants undermine vaccines?
Sorry, say again?
Yes, the question was the new variants, the new COVID-19 vaccines, the variants, can they undermine vaccines?
Yes, they can undermine vaccines. Of course, vaccines are still working well against protection from severe disease and death, but we should not take that for granted. So we should still attempt to control virus and expand vaccines as quickly as possible in order to prevent further evolution of the virus. That's the thing we worry about.
So you mean that vaccination or no vaccination, the otherwise infection prevention principles should still be very strongly observed?
I believe so.
Okay. Right now, I do not have any questions here. Viewers, if you have any question, you can unmute yourself and ask your question directly to Dr. Gupta as well. I feel we still have a little more time, so you can go for it if you so wish. Dr. Vaidya asks a question, Dr. Pierre Vaidya asks a question, how do they calculate the R values?
So the R values are calculated by epidemiological data. In other words, the number of new infections recorded of the infection. So that's how they're calculated. They just look at but it's more complex than that. There are a number of different parameters, and I'm not an expert in epidemiology, so I don't know exactly how they do it. But it's based on the number of new cases over time, and fitting a modeling distribution, a model to it.
Dr. Gupta, you talked about Delta-specific vaccination. Now, I heard about the cases where under CEPI and similar some organizations, they were planning for a 100-day vaccine project or the Disease X project, where very quickly a vaccine can be developed for some new variant, some new virus altogether, new pathogen altogether. Is there any effort on already towards having a Delta-specific vaccination?
I have heard that the AstraZeneca people are thinking about it or actively trying, but actually I've not seen anything solid yet.
And
so I believe that people are.
Oh, okay. I'm sorry.
I think it's happening, but there's little information available at the moment.
Okay. And what is your opinion about this case? Because there's a lot of discussion in India about the fact that incomplete vaccination and slow vaccination can result in more variants coming up. Is that a possibility? And in that case, what could be the solution?
Sorry, the solution for
there is a discussion that slow vaccination or incomplete vaccination or partial vaccination can result into more variants and more unknown possibilities and levels of transmissibility and infectability. So what could be the case? Is it real? Is it really a possibility? And if it is so, then what could be the solution for it?
Yes, I think that yes. I mean, we do want to comprehensively vaccinate if possible, because allowing, as I showed you, transmission to happen even in partially vaccinated population, the virus will still be able to cause chronic infection in people, as I showed you, and that's the ideal training ground for it to evolve new mutations to confer more escape and more mutations. So we want to avoid that. So countries should be vaccinating as quickly as they can, even if numbers of cases is low. I mean, that's especially when you want to vaccinate when cases numbers are low.
What is the current situation in UK? Is it all under control, or you are really concerned that this Delta can create further spikes there?
In UK, we had quite a high acceleration of Delta, and the number of cases is quite significant. The case numbers may be falling slightly, but we think that this wave in the UK is set to be here for some weeks to come.
Is it the same thing with India?
I think in India, well, we are seeing rising cases in a number of different places. So yes, I think it's happening there too.
Now, is it happening in pockets, or it is very general? It can be seen throughout the country.
I haven't been looking at the sort of specific regional data myself, so I'm not best placed to know that, sadly.
There is another question here, and the question says that the Delta variant of the coronavirus was primarily responsible for the second wave of COVID-19 in India, according to Dr. N.K. Arora, co-chair of the Indian SARS-CoV-2 Genomic Consortium. It is
Yes.
now considered as the dominant strain. Which strain can cause the possible third wave in India, in your opinion?
I think the third wave is most likely to be a Delta again.
Okay.
Yeah, because that's the dominant strain now globally, and it has significant advantages in transmission and immune evasion. So it's still the dominant one. There may be a variant that comes up on top of it. We thought about this sort of the Delta Plus, but that didn't really seem to take over. But something similar to that could emerge.
And in your presentation, you also said that the so-called double variant is not that much something to think about.
Well, the double, yeah, that 0.1 virus has 2 mutations, but it does not appear that the second mutation is adding is synergistic. In other words, it's not doubling it's not adding up together. It's just modulating the immune escape, and that explains potentially why 0.1 was not so much has less immune evasion or similar immune evasion, but is much more successful because for some reason it's more infectious.
Okay, we have one more question from Sri Shiva Prasad Kenneth, who was one of our senior directors, and he asks, there are several studies of infection spread and morbidity in India, which are in variance with what the officially stated data, and the Delta variant, it is said it is more infectious. What is your understanding of the spread of infection and morbidity with the Delta variant?
Yeah, I think that it is more infectious. We pre-printed a piece of work that suggests that the success in India and the growth rate of in India was driven by immune evasion plus increased infectivity. So yes, absolutely. The viral loads do appear to be higher as well in people with Delta compared to other viruses, and potentially the generation time, the time period between one person being infected and the next person is shorter. In other words, the virus is jumping from one person to the other quicker, which is a very worrying thing.
Okay, so right now I do not have any more questions, Dr. Gupta. It was so very wonderful for you to join from your busy schedule, and thank you so very much for this program. Thank you very much.
Thank you.
Thank you. So viewers, here we end up the program today, and we will come up once again with more issues, more concerns, and look into it with the help of our experts. And we sincerely thank Dr. Gupta for taking his time and joining us. Thank you all. Thank you all for joining us in